I want to take the pleasure of introducing today's speaker. This is a person who I care very much about and know very well. He's very gracious to come and visit our campus from far, far away. And so we appreciate it. Dr. Gautam Chowdhury is a distinguished professor in the Department of Obstetrics and Gynecology at UCLA, where he serves as executive chair of the department. And the executive chair means that he actually is a chair for a number of different branches that are affiliated with UCLA. He's also a distinguished professor in the Department of Molecular and Medical Pharmacology. He completed his MBBS from the R.G. Carr Medical College in Calcutta, and then went on to do his MD at the All India Institute of Medical Sciences in New Delhi. His PhD was completed at London University under the tutelage of then Nobel laureate Sir John Vane, who's since passed away. His residency was then completed in the United States. He came to the United States after his PhD at London University to pursue a residency in obstetrics and gynecology at sunny Buffalo. And from there, he went to do a fellowship in reproductive endocrinology at UCLA, where he has remained ever since those young years that he joined UCLA. Dr. Chowdhury has made a number of groundbreaking contributions in the field of reproductive biology, of which I am part of, really primarily focusing on the role of bioactive substances in reproductive pharmacology. For example, he did a number of studies that actually brought together the unifying theory that actually IUDs, intrauterine devices, really work through the activation of the prostaglandin pathway, and that is their main mechanism of action. Now, Dr. Chowdhury has received lots of national and international recognitions. Today's is just one minor part of that. He received the Rambaxi Award for Medical Research in 2006. And the President's Distinguished Research Award from the Society for Gynecologic Investigation in 2010. He has served on the Advisory Council of the National Institutes of Child and Health and Human Development, and the main, one of the large institutes, obviously, of the NIH. On a personal basis, Doctor, I don't know why, is this changed for some reason? I have to go back, I guess, huh? I'm messing up your presentation, Gautam. There we go. On a personal basis, I really did want to share with you a couple of things about Gautam Chowdhury if you don't know him yet. He is obviously a researcher par excellence, extraordinarily well trained, very well connected. But more importantly, he is truly one of these individuals who embodies much of what is good in academics. He ensures that a young generation of individuals receives the best and the best protection. His faculty, his young residents, postdocs, and so on really pay high tribute to him because he ensures that we have that next generation of individuals. He also tries to ensure that it's a fair process. You know, often academics is a dog-eat-dog kind of world, and he protects those that need to do so. So he is actually, in addition to being a great researcher, he's a great human being, a great academic, and we have much to learn from him. Gautam, thank you for coming and visiting with us.
Thanks. Thank you very much, Ricardo, for inviting me here. It's been a great pleasure always to come and visit here, because every time I come, I feel it's like it's a jewel hidden. And I know you've been trying to propagate in a very good way. In fact, in our field of obstetrics and gynecology, some major contributions were made from this institution, and Dr. McDonald is here, who was part of it, with a big team of Robert Greenblatt, Mahesh, Virendra Mahesh, Somnath Roy, and obviously Dr. McDonald. And Somnath Roy just passed away very recently, so it's been great. When Ricardo asked me to give a talk, I initially thought I'd give a talk on the cardiovascular system. But knowing about the Pollocks being here, and Dr. Cottrell was there, I thought it would be embarrassing to talk of cardiovascular topics here because, as you probably know, that this is one of the greatest centers for cardiovascular research, and not just coming from me, but from Sir Salvador Moncada, who indicated that he came for a visit, I think about a year ago maybe, and said 2 years ago, and said it's one of the best centers he's come across and enjoyed it, and I hope he'll visit again in a way. So I'll be talking on free radicals and their implications, breast cancer. Ricardo said I can't talk on any published topic because then people will just feel that it's an old. So I've put some slides, they're not completely in order, so excuse me, number one, and also realize that I'm running 3 hours behind time, West Coast time, so I have to get up 2:30 in the morning, LA times, to be in time for the grand rounds. With that said, I need to explain, and I'm sure there'll be some junior people here who are doing studies, so for them, it is interesting now, I'm a gynecologist. I don't take care of patients' breast cancer, and as Dr. Katravas and the Pollocks know, that I've been actually interested in vascular biology. Why did I suddenly switch to cancer and breast cancer? It's kind of an interesting story. I had a patient, and I was doing an annual exam on her, and she says, "Oh, Dr. Chaudhary, it's my 50th birthday, and I want to do something good for academia, so what is the chancellor's name and address?" I said, "What do you want the chancellor's name for?" Says, "Oh, I want to give some money instead of holding a party." I said, "You don't give to the chancellor, you give to the department." Sorry, Ricardo, nothing against. Ricardo has been more than a brother to me, and I miss him, so I can take a front on that. But this is true. So she says, "Yeah, I'm interested in breast cancer, and you don't work on breast cancer." And I said, "Oh, I've always been interested in breast cancer." But unfortunately, I had no money for it. So she says, "Oh, in that case, I will provide funds for you." And with that, I got preliminary data, and apparently I was told that mine was quite many years ago, the first grant on nitric oxide in breast cancer. So that's how I got into breast cancer, and it's filled a productive team. The other thing, what is interesting is, I was trained as a classical pharmacologist, where in this morning's talk you saw we did bioassay. When I did work on prostaglandins, we didn't have radioimmunoassays then. So it was all bioassay, and John Wayne taught us all the bioassay aspects. So basically, when I came, and now with the changing things of grants, you have to do some molecular biology work anyway, the dots and other things. And then what I learned very fast is that it's a two-way street with postdocs, that they learn, I don't know what they learn from me, but I learn a lot from them. And one of the things is learned a lot of chemistry, although I'm not good at chemistry, and I was very bad. That's why I became a physician to start with. But so it's been a collaborative effort, and this work is done by one of my postdocs, Sivajit Sen, so I put his name first, about the breast cancer. Now, what is interesting is I know that he had indicated that Moncada is doing on mitochondria. What I'm going to do today is just talk very briefly about one of the hypotheses which people have been thinking about, the differences in the mitochondria and other functions, and then free radicals in breast cancer, and role of superoxide reactive oxygen species in breast cancer, and role of nitric oxide and its interactions with superoxide and reactive oxygen species in breast cancer, because we had to bring nitric oxide in to justify my existence. Now, it's thought that mitochondria was actually when a bacteria, a prokaryote, entered to the cell, and this is a diagram of a prokaryotic cell, as you know, and what is kind of interesting, I think I missed a slide now, is that a fascinating thing which we are working on, and I'm going to pass on a CV of individual, Gautam Dhar, who works with me on a fascinating work which we are doing. And what is interesting is that we have the glycolysis in prokaryotes. Prokaryotes, they became eukaryotes after they entered a cell. And this works, generates ATP through the mitochondria by oxidative phosphorylation, whereas prokaryotes is by the method of glycolysis. Cancer cells, so eukaryotes, when they transform, and I'm not going to go into the controversy of stem cells, but eukaryotes, when they become malignant cells, again revert to glycolysis. So that's and then generate ATP, although mitochondria will put something in. And then if we look at another fascinating thing, that you have embryonic stem cells where glycolysis is a major part of ATP generation. These cells undergo differentiation, and then it's the mitochondria that is generating ATP. And then when we become malignant is glycolysis and ATP generation. So it's like a cycle in a way. And at the end of the talk, I'm going to indicate some work which we have not yet sent. We are finishing some finishing touches on this very exciting aspect. So here's the mitochondria. You have the outer membrane, inner membrane, as you know. And the reason I stress on, and this is the electron transport system of the mitochondria. And the reason I indicate is in non-malignant cells, there is some generation of some superoxide radical, which with the cellular superoxide dismutase gets converted to hydrogen peroxide, and this is inactivated by antioxidant enzymes. Whereas in the ATP synthesis occurs by oxidative phosphorylation. When we look at the malignant cells, however, what is interesting, there's excessive production of superoxide. Superoxide dismutase is really increasingly expressed. It increases hydrogen peroxide, and we and many others have shown that hydrogen peroxide acts as an endogenous growth factor. And just so that if you've had your lunch and fall asleep, just to indicate to you that hydrogen peroxide, what we have shown is inhibits PP2 activity, and that prevents the dephosphorylation of AKT, and therefore cells continue to proliferate, and there's decrease in the antioxidant enzymes. So I'm going to focus on this particular aspect today. Now, as I said, reactive oxygen species is produced in significant amounts by mitochondria as a byproduct of oxidative phosphorylation. Damaged mitochondrial DNA preferentially the reactive oxygen species do, and mitochondrial DNA mutations cause defects in mitochondrial respiratory enzyme complexes and increase. What is interesting, there is a person, Keshav Singh, he's in Alabama now, was in Roswell Park, where he's done some fascinating experiments, where he's taken cancer cells, taken the abnormal mitochondria out, and put in normal mitochondria, and they behave. They at least revert quite an extent to actually normal cells, which shows that mitochondria, at least in certain circumstances, may play a very important role. What is further interesting is mutations in mitochondrial DNA is found in most cancers, including breast cancer and ovarian cancer, and depletion of mitochondrial DNA in human breast epithelial cells is done, resulted in an in vivo tumorigenic phenotype as well as development of tumors in vivo mouse model. This was again by Keshav Singh. So mitochondria plays a very important role, and the superoxide source in cancer cells is most likely just oxidative phosphorylation and NADPH oxidase 1. And what I learned from Suvajit is that superoxide gets converted by superoxide dismutase to hydrogen peroxide, and it's interesting that the main enzyme involved in manganese SOD, and this is actually in the mitochondria itself. And hydrogen peroxide is formed, and in the absence of free metals, the catalase or pyridoxine or glutathione peroxide ones will inactivate to water. Now, what I'm going to show you in some of the following slides, and this is the hypothesis that we've shown, and I'll come to it, that the expression of the antioxidant enzyme decreases in cancer. This leads to an increase in hydrogen peroxide, and this then acts as what we think an endogenous growth factor. Now, there have been some studies, which is interesting, by Oberle's group, where they show that superoxide excess, when they've transfected and increased the MnSOD only, that high amounts of superoxide are formed, and that also leads to cell death. So what is happening probably is hydrogen peroxide has a bimodal action. What we show, as you will see later, is low concentrations when you transfect and low, with antioxidant enzymes, hydrogen peroxide level goes down and cells will go under apoptosis. In intermediate levels, they cause proliferation. We've not looked at the higher amounts, but other investigators that add high amounts of hydrogen peroxide exogenous will also undergo cell death. So what happens is cell death could occur at very low levels in cancer cells of hydrogen peroxide, and higher levels. And intermediate levels will cause proliferation. And I'll later come to some of the clinical implications. What we used is use certain cells which are well-differentiated cells in the breast cancers, and we use a highly undifferentiated cell line, MDMB231. And basically, we use as a control human mammary epithelial cell line, breast cancer, human primary breast epithelial cells, and also we use MCF10A. And the reason why we call them normal is that if you look at the normal cells versus cancer cells, what is interesting is the milk protein casein kinase is expressed much higher in normal cells compared to cancer cells, which again indicates that as the cells undergo undifferentiation, that the ability for milk proteins is gone. What we did is measure the superoxide levels in normal cells, versus this is HMAC cells, I think, versus the cancer cells. And there's no question that the superoxide levels are significantly increased in the cancer cells. And that's not unexpected, and other investigators have shown that to be the case. We used a technique which was initially described by Cullen Raman at Wisconsin, where he measures the 2-hydroxyethidium, and that's actually considered now by many as one of the main methods for measuring superoxide. And then, just to show you again, is some other slides showing the normal breast cells versus MDMB468 cells, and again, superoxide is increased. And this shows a whole area of cell lines where we take MCF10A, which is a normal cell line, slightly produces more than the basal, but all others, cancer cells, are significantly produces greater hydrogen peroxide levels. And again, comparison in tumor versus normal breast cells is again the same aspects with numerous experiments. Now, when we look at the magnesium superoxide dismutase in human breast epithelial cells, please note that this, as I indicated, is expressed higher in the cancer cells, which allows for more conversion of the superoxide radical to the hydrogen peroxide. Now, what is fascinating is when we looked at the antioxidant enzyme system, we find on the left-hand column here is MCF10A. Look at the antioxidant enzymes, and they're all, in a way, this is GAPDH, and antioxidants are decreased in all, except what was interesting, we found by Western blotting that catalase expression by Western blotting is increased. But when we looked at the bioactivity of the catalase enzyme, it was decreased. And this was not surprising because we looked at some of the literature by Erwin Friedrich many years ago when he published, is the superoxide can actually inactivate this catalase enzyme. And if we later found that if we put in antioxidants, we could actually revive the activity. So it's actually superoxide that is doing it. And this, again, sometimes shows the fallacy of just doing Western blotting to interpret results rather than looking at bioactivity as well. And I know the cardiovascular group, they always look at the bioactivity here. It's all their lab. So that's kind of important. And I'll come to this catalase later on again. And as you look at it with the expression of normal breast cells again, human mammary epithelial cells versus the other cell lines, this slight increased expression. But as I said, the bioactivity is actually reduced, which is something no. And again, we show with different cell lines, again, the HME cells and MDMB468 and MCF. The last two, actually, this and this are the cancer cell lines where it is diminished. Now, when we look at the hydrogen peroxide levels, here is a very interesting thing. Hydrogen peroxide levels are increased, but when we actually overexpress catalase, the catalase level goes down. And this shows the increased catalase expression. So what is then happening is that obviously the catalase is normally not functioning well, and when in the cancer cells you express, you can bring catalase levels down, and after a certain level, the cells undergo apoptosis. This is here is the catalase again in overexpressed cells. You can see the hydrogen peroxide levels going down, and you can see the PARP and others, going down in the overexpressed cells, showing again that the apoptosis is occurring. And then when you overexpress with glutathione peroxidase, same thing happens where you have the overexpression here and cells undergo apoptosis following overexpression. Now, what we also showed is that catalase overexpression can act, what happens is it inhibits the, as I said, high hydrogen peroxide levels inhibits the PP2 activity. So when we overexpression of catalase, it decreases CO2 and the levels will actually go a bit up. Now look at this particular slide, just the last one, where we use PEG catalase, and then we show that effect of catalase bioactivity in the absence and presence of antioxidant superoxide dismutase. And when we give PEG SOD, again, the levels of the biocatalysis activity goes up again because the antioxidant, because now the superoxide is being taken care of. And normally, as I said, superoxide would inactivate the catalase enzyme, but when you use SOD, this actually helps out to increase catalase. So when we looked at, again, the catalase overexpression changes, you can see that the fold change in specific activity, mechanism of apoptosis and MBM4 upon catalase overexpression, we find increase in the BAX and other proteins with decreased phosphorylation of the AKT and ERK, which again shows, as I indicated, is that the PP2 activity is actually hydrogen peroxide inhibits it, and therefore normally they would be phosphorylated, remain phosphorylated, but when you remove it by overexpressing catalase in these cells, hydrogen level peroxide goes down, the PP2 activity is then increased, And therefore, with the phosphatase increase, you get dephosphorylation of these enzymes. So what is interesting, then you would probably say that, is this true in humans? And we had 4 samples in humans. And again, what was interesting, I don't have the Western blots here, but Western blots were about the same, or if not more. But when we look at the bioactivity in the tumor tissues of human specimens, the catalase activity has gone down again. So it might suggest that catalase decrease in activity, as we had indicated, may lead to increase in hydrogen peroxide and increased growth of breast cancer proliferation. Now, what is interesting is there have been some studies done where women have been given estrogen and their breast catalase, those who develop cancer, they actually had a single nucleotide polymorphism of the catalase enzyme. So it is possible that catalase underexpression may be an important part to play with relation to the human breast cancer model as well. Now, where does nitric oxide come in? And this is, again, a very, very interesting thing. The first, actually, study was shown by Sir Salvador's group, where they showed increased expression of INOS in breast cancer. And then there'll be some other studies being done. And what is interesting is very recently, about a year ago or so, David Wink from National Cancer Institute showed that expression of nitric oxide correlates with the degree of undifferentiation, and the greater the degree of undifferentiation, the greater the NOS expression, and worse the prognosis. So in estrogen receptor negative breast cancer cells, if they express nitric oxide synthase, then the prognosis is worse. And the question comes, what is nitric oxide actually doing then to help in the breast cancer cell proliferation? So, and as I said, increased NOS predicts poor survival in breast cancer patients. So in multivariable survival analysis, it has been shown that increased NOS2 levels influence survival, predicted inferior survival in women with ER alpha negative tumors, and exposure to NO also enhance cell motility and invasion of ER alpha negative cells. So these two would suggest in some ways that nitric oxide is involved. Now, quite some years ago, we had, and this was published, is we looked at the nitric oxide expression in various cell lines. And you can see here that in some cell lines, and this is important to note, is ZR7530, and in BT474, the cells have a somewhat greater expansion, as is MCF12F. Now, what is interesting, these 3 cell lines will come into it, which is fascinating, interesting. So please note that in the studies of nitric oxide here, we use 3 cell lines, ZR7530, which is a cancer cell line, MCF12F, which also produces nitric oxide, but is a benign human breast epithelial cell line. And then we have BT474, which produces somewhat less than MCF12F, but this is also a cancer cell line. So ZR7530 is a cancer cell line, BT474 is a cancer cell line, MCF12F, which produces also nitric oxide, has nitric oxide synthase activity, is a benign cell line. And then we have this 2 other cell lines, MDMB231, highly aggressive, and MDA-MB468, which has very little. So for some of the studies which you'll see, we use these 2 cell lines, MDMB231 and MDMB468, and later on to see the bioactivity of endogenous nitric oxide, we use these 3 cell lines. what we found was fascinating is we used the NO donor, and as you know, NO has a very short half-life. So we used the NO donor, which has a constant release called delta-NO to resemble a constant release of NO. And what was fascinating to us is low concentrations actually caused an increase in proliferation followed by apoptosis. Now, what happens is when we submitted grams, they would say, my God, why are you using millimolar? But please know that millimolar is not the amount of NO, but the amount of the compound, because we had shown that it releases NO in micromolar up even to nanomolar, picomolar concentrations, because this is a 24-hour release. And what we observed then is there's an increase in cell proliferation with low concentrations, say 30 and 60 millimolar, but they're low picomolar to concentrations, actual NO concentration, but then it goes down. And if you look at also the other factors of thymidine uptake and all, this shows a bimodal action, low concentrations increasing and high concentrations leading to apoptosis. And the same is effect of N on protein synthesis, again in the 30 and 60 micromolar concentrations, that's the actual levels of NO released from that NO. We find that there's an increase in protein synthesis, and this is a concentration response related curve. So low concentrations produced NO can actually lead to increased protein synthesis and proliferation. Now, with that, these were exogenous NO. And what is fascinating, we then started trying to see what we can do with cells that produce endogenous nitric oxide, because at the end of the day, that's what is going to count. Why is NO expression leading to increase in cell proliferation? So what was fascinating is, remember, we showed about the NO production early on, but we separate the mitochondria, and we found that these cancer cells, it is the mitochondria that is forming, has the NOS activity and producing NO, but not MCF12 cells. So one is the site, and therefore this is important as we focus on what is the NO produced from the mitochondria, how is it leading to proliferation, and as this is not produced by the mitochondria. So this then made us focus on probably the action of NO, endogenous NO, on the whole aspect of mitochondrial. So what we then did, just to show is NO involved, is fascinating in a way, that we used L-name in very high concentrations. And as you can see, that NO production was, as I said, decreased. And when you had L-name plus arginine, it reversed. So nitric oxide inhibition actually caused a decrease in the cell proliferation, which is kind of very interesting. Then we looked at the proliferation rate at different groups. As you see, there's the rate of proliferation was also, again, concentration dependent, where L-name, as you can see, has the control. L-name can inhibit in a concentration-dependent manner, the rate of proliferation. So what we then can surmise from the L-name experiments is nitric oxide allows cell proliferation. And if we inhibit it, the cell proliferation stores. The question is, what does it do to increase the cell proliferation rate? Now, I'd just like to show you this, this slide, where again we have control cells, L-arginine alone. We have 5 millimolar with a very high dose of L-name, decrease. You give L-arginine, it prevents the effects of L-name, which is well known. But when you add ODQ, a guanylate cyclase inhibitor, there is no effect on the proliferation, unlike nitric oxide. So this then told us that this is a cyclic GMP-independent action of NO and not a GMP-mediated, because in nitric oxide, people will say they said GMP-dependent or independent. So when we looked at the signaling pathways again, you can see that with L-name on the right-hand column, the phosphorylated AKT and the ERK are all decreased compared to normal cells, and that's not unexpected. And what is also interesting is, I don't know whether you can see it clearly here, but the S phase in the L-name areas, here is about 20, and here is about 11 or so with L-name. So again, the cell cycle is affected as well. And we took the other cell line. The one was ZR7530. This is BT474. Again, same effect, inhibition. And these are the two cancer cell lines which has nitric oxide being produced in the mitochondria. And cell count is also decreased. And even, just to tell you, even the cell cycle is decreased from 22 to about 11 S-phase cells. But when we looked at the MCF12F, and this is a benign cell line, which still produces nitric oxide, but not in the mitochondria, L-name did not actually influence its rate of proliferation. So how is this site of action where NO is produced leading to proliferation? Again, the L-name on viable cell count is diminished. And same with regard to 12F, this L-name had no effect on the S phase, basically. They were the same. So I'll just briefly mention that what happens is, as you know, they all are producing electrons here. But nitric oxide, and this was again shown by Salvador Moncada, the nitric oxide has a greater affinity for cytochrome C oxidase compared to oxygen. So when you have nitric oxide being produced locally in the mitochondria, it is combining with the cytochrome C oxidase, so the electron transfer is affected, and we show that what is interesting is hydrogen peroxide is increased. So nitric oxide, and that's the interaction between hydrogen peroxide and nitric oxide. So that the hydrogen peroxide normally will cause proliferation, nitric oxide further accentuates that because it combines with cytochrome C oxidase, and the electron transport chain system is affected, hydrogen peroxide is increased. And this one, we show that the cytochrome C oxidase activity is in before treatment with L-name, after isolation of the mitochondria, and this is following treatment of the cells with L-name, again showing that cytochrome C oxidase activity is increased, again indicating that NO was binding to cytochrome C oxidase. And L-name, as we show, as I indicated, because nitric oxide is now reduced, we also show simultaneously the hydrogen peroxide levels are diminished. And this will account for the low rate of proliferation with L-name. So again, what we show here is a beautiful interaction between a nitrogen reactive species and the reactive oxygen species. So reactive nitrogen species is not acting just alone, but also in combination with the reacting oxygen species. And we think that is acting through hydrogen peroxide. And again, when we look at L-name, I'm sorry of the color of the slides. I like to tell my secretary who got over-enthusiastic, is that the S phase here is decreased in the L-name from about 16 to around 5, whereas with ODQ, there was no change again supporting that concept. And when we did either the overexpression of catalase or glutathione peroxidase, again, the S phase was significantly diminished, as you see, from 20 to 11 and all before the cells died. So this again shows the interaction of these enzymes. Now, one of the things which I will just very briefly mention without showing the data, because we're still in the process of doing some more experiments, is fascinating, is, one of the things is nature is kind of fascinating and beautiful. And we said that cancer cells need more ATP, and we all agree to that. Our glycolysis is one of the most inefficient manner of producing ATP. So why, and nature is much more intelligent than us. But the general concept is the Warburg hypothesis in a way that cancer cells are glycolytic, and there's no doubt that that may form a part because glucose uptake is increased in cancer cells. But in most cases, you cannot account for the rate of proliferation with the amount of glucose uptake. So there must be some thing. So actually, and I'm not a chemist. So what I told my lab people, and Gautam Dhar is an adjunct assistant professor at UCLA. He has a very, very good chemistry background, and he hadn't worked on cancer. He works on DNA repair. So there were some issues in his previous lab where he wasn't being allowed to be independent. So I said, come over, work on the mammalian cells, because he was working E. coli, and let's do something together. So I said, we should have a project which is high impact, but may never work out, and let's do some other work which leads to publications like we all do. So we then thought and put in, and a fascinating thing came to us, is this is the normal way in mitochondria that the pump is in the proton gradient releases ATP, as you know, from ADP, and the electron transport change in the inner membrane. And what we now think is in the cancer cells, I had a slide which probably disappeared, is what we think is that ATP synthesizing apparatus has now gone to the plasma membrane. Why do I say that? What is fascinating is that the extracellular milieu of cancer cells is always acidic, but the intracellular is more alkaline. And under those circumstances, what people have done is they've used carbonic anhydrase inhibitors, and the cancer cells start shrinking because the outside acidic milieu is no longer there. So what we think is happening, and we have some evidence, is that the proton gradient in the extracellular fluid, now that the ATP pump in cancer cells, which we think is on the cell plasma membrane now, allows for the influx of the proton, and that turns on the ATP synthesis. The only thing which we are having trouble explaining is how's this proton thrown out. If it needs energy, then it's a futile cycle. You're bringing in energy and using energy to extrude it out. So we looked at all the transporters, but it's difficult to explain how against a concentration gradient, proton can be released, but science is not advanced to that extent. But it's fascinating. We've shown that you can't explain the glycolysis, just ATP synthesis in cancer on the basis of glycolysis alone. So this is something fascinating, and Gautam Dhar is working, and I'm going to pass on his CV, because chemistry is so good, and I didn't want him to rot in my place there. So in conclusion, excessive production of superoxide leads to human breast cancer cell proliferation, and superoxide and nitric oxide can react to cause breast cancer cell proliferation, but can also cause apoptosis, which is concentration dependent. Now, when you come from LA, some people ask, oh, you must have rich patients. And we know Alan Descherney, we were talking about his Hollywood patients today. So, I come up for 5-year review. All chairs have 5-year reviews. And my last review, the only criticism was, thanks to Ricardo, who helped praise me, is that I've done no fundraising. And I said, I don't know any rich patients. So Jonathan Barrack, who was one of the vice chairs, now chair at Stanford, he knew Nicole Kidman. So he said, oh, there's a free table for some fundraising. Do you want to come? I said, okay, I will be there. So I went to that, and here I scrambled to get next to Nicole Kidman here, pushing everyone. Here is Barrick here, his wife and daughter, me, and then Larry Platt and Robin Eisner. And then I went to my secretary after the photograph and said, cut everyone off. And then I sent this to all my friends and things, saying, with love from Nick and Gautam. And trust me, my reputation, just knowing Nick, I became more popular than ever as a teacher or a clinician or as a scientist. Then I knew I did choose the wrong profession anyway. But then it lasted for some time till people found out, and I'm back to square one again. Anyway, thank you so very much for this opportunity. And just to let you know, I'm very, very grateful to Ricardo for having me here. He's missed by many people, as you know. And we talk quite frequently over the phone. But, you know, Ricardo, you've been doing great for this institution, putting the names. You have jewels in the cardiovascular, I know. You're trying to develop the cancer-related institutes. You were actually in charge of the cancer thing. OBGYN made great names here, and I'm sure other departments do as well. So I look forward, hopefully, if invited again, coming back, and thank you again for your hospitality, which is just great.
I think we may have some time, thank you, for questions.
Sure.
And we have some here.
I do have, I would like to start out with a question. That is a disturbing photograph. Tell us a little bit, you know, we are beginning to focus much more on translation, which we have some, but not as much as we'd like. Tell us how you would envision, obviously your data is not complete yet, the storyline is developing, how you would envision this proceeding into a translational path. What ideas have you been able to generate in that?
Yeah, well, that's a very interesting question. And, you know, there are some data in animals, actually, where they've shown that with antioxidant enzymes given to animals and rats, that the breast cancer incidence is diminished, which is fascinating. We don't know whether one can give selective antioxidants to humans or not. We are trying to screen some compounds. But I think in general, you know, we'll have to look at it. But from a pathophysiology point of view, for example, there's a study which was done in New York, as I said, where the single nucleotide polymorphism of the catalase gene did lead to more incidence of breast cancer in women on hormone therapy. So one would be perhaps people who are being screened to look for this single nucleotide polymorphism. If not, they're at a lower risk, we can do that. And, you know, the antioxidant studies, you know, I don't know, people, I just saw last night on television about some big pumpkin where they say is good for women's faces because it has high antioxidants. Maybe some dietary factors will come in, which could be important. I just don't know. But that's the trend, I think, which we will go to. Jennifer?
Nice talk. I'm not a cancer person, but
I wasn't as well, you know.
But I was really intrigued by the difference between the cell lines that were benign versus the malignant ones and the responses with the L-name with proliferation. And I was just curious if you'd looked at any of the transporters, because either L-name not getting into the cells or just availability or the actual activation of NO within endogenously in the cells.
No, that's a very good question. And basically, the answer is not to do with transporters because, you know, one of the slides was missing where in the MCF12F cells, we show there that the nitric oxide synthase NO release is going down with L-name. Okay. But it doesn't affect the proliferation. And it's not in the mitochondria. Now, I know that, you know, there's a person who was publishing, he was in Ohio, Guzbelain, I forget his name. But he's been published mitochondrial, and Salvador and I visited there once. And one of the things is mitochondria may not synthesize NOS. We don't know whether they have but whether it was transported there or not. So we don't know that. And maybe that's the problem, where it starts, where the NOS gets transported into the mitochondria. But no, NO is produced there. We show that alanine does decrease, but it doesn't. And the reason is it's not produced in the mitochondria. So the closeness of it is affecting. And the other NO is probably diffused and inactivated by the other systems.
That's interesting.
Hi, how are you? Let's see, I'm a radiation oncologist, so I thank you for making that transition and saving our field. Let's see, what I'd like to ask you is related to what Dr. Aziz asked you about the translational component. Now, if you were to use radiation therapy, for example, which is highly O2 dependent, and hypoxic cell survival is much greater than in patients undergoing radiotherapy, what would you envision for the combination of one of these inhibitors, perhaps, or the manipulation of the system in another way, perhaps?
Yeah, it is interesting. You know, it's interesting when we talk of low and high concentrations of NO. We have, rather when we say we generalize, there's a person in UC Santa Cruz who's sending us some NO material. We have suggested before for breast cancer that one of the mechanisms could be is where we tag on, say, tamoxifen to a NO donor. Then it will be distributed in ER-positive cell lines, cells, and then the cells can undergo, high concentration of NO can undergo apoptosis. David Wink has shown, actually, it's a fascinating thing, just exactly opposite to what we're talking, is they think that NO irradiation may be made more sensitive by nitric oxide. And we don't know the exact mechanism, so we still have to know about that. But I know that people are trying NO and radiation, and they think that high amounts of NO may actually increase the sensitivity of cancer cells to this. Just like we've shown that high amounts of NO cause apoptosis. Please.
So thank you very much especially for combining cancer and cardiovascular biology. They have a lot of things in common. So you really had two talks today. One was for the nitric oxide, and the other one was for how different malignant and normal cells handle ATP or produce ATP. And it brought to mind, there is a group in Alberta, Canada, that they believe exactly the same thing that you're showing, and they have shown that by using dichloroacetate, DCA, which is a cheap, you can call it orphan drug, there is no patent on it, they can beautifully block the progression of astroglioblastoma. And they have used it in patients, terminal patients, and they have seen amazing results. So, and the idea behind this is, as you talked about cell proliferation, cancer cell proliferation. So the lead scientist is a cardiologist. His interest is pulmonary hypertension. And he was trying to find something to block smooth muscle cell proliferation for pulmonary hypertension. So he went and says, well, let's go to something that's really more dramatic than the long-term pulmonary hypertension. Went to cancer, showed this. This was published in Cancer Research. He has been on Newsweek. And now it turns out to be even more useful for pulmonary hypertension patients. But it's an amazing story.
And you're right. You know, this nature, everything is common.
Yeah.
We just look at different systems. And therefore, having worked on the cardiovascular is just using breast cells and putting it together, and that makes something unique with the NO. Nothing, but cytochrome C oxidase was shown, I think, by Cellular in the vascular system, not in cancer cells. And when we apply it, it applies even to cancer cells. So you're right, that science, it doesn't matter what system you're using. Ultimately, it can be in any part. Thank you.
This is a plaque that will read, to Ganam Chowdhury, MD, PhD, in appreciation for his leadership in nitrous oxide research, women's health, the mentoring of young leaders, and for his participation in the 2011-2012 presidential lecture series.